My name is Wong Jingling. My matrix number is AA200758. Now I'm going to talk about microscope. Before I start my presentation, I'm going to introduce a little bit about microscope. First, what is microscope? Microscope is an instrument used to see objects that are too small to be seen by the naked eye. There are many types of microscopes and they may be grouped in different ways. But for this video presentation, I'm going to talk about a light microscope. A light microscope is a biology laboratory instrument or tool that uses visible light to detect and magnify small objects and enlarging them. Function of a microscope is they use lenses to focus light onto the specimen, magnifying it, thus producing an image. The specimen is normally placed close to the microscopic lens. Now, I'm going to talk about the function of each part of microscope. First, eyepiece. Eyepiece is a part of an optical system which is directed to the user of the microscope. It contains the ocular lens which user looks through to see the magnified specimen. Next is lens tube. Lens tube hold the eyepiece and connects eyepiece and ocular lens to the objective lenses which near the microscope stage. Objective lenses. Objective lenses combine with the eyepiece lens to increase the magnification level. Microscopes generally features 3 to 4 objective lenses, with magnification levels ranging 4 times to 100 times. The next one is nosepiece. Nosepiece contains the objective lenses. Microscope users can rotate this part to switch between objective lenses and increase the magnification power. Microscope arm connects the eyepiece tube to the base. This is the part where the user holds when transporting a microscope. Next is microscope base. Microscope base provides stability and support for the microscope. The next part of microscope is illuminator. Every microscope requires light for viewing. This can come in the form of a low voltage illuminator or a mirror that reflects external light source such as sunlight. The another one is stage and stage clips. The stage is a platform for the slides which hold the specimen. The stage typically has a stage clip to hold the slide of the specimen firmly in place. Another part of the microscope is condenser lens and diaphragm. The condenser works with the diaphragm to focus the intensity of light onto the slide containing the specimen. These parts are located under the microscope stage. This is a part for the method to handle a microscope. First, we should hold the microscope with one hand around the arm and the another hand under the base. This is the most secure way to hold and work with the microscope. The second method to handle a microscope is avoid touching the lens of a microscope. This is because the oil and the dirt on our fingers will scratch the glass of the microscope. Third, we should clean the smudges on the microscope glass with a piece of lens paper. Avoid using other materials such as cotton. This is because they will scratch the microscope glass too. Fourth, rotate the nosepiece to the lowest level after finish using the microscope. If we leave the nosepiece up while storing it, the gear on the device will wear out. Lastly, you should cover the microscope with a dust cover to protect it from dust and debris. Now I'm going to share about the method to use a microscope. Step 1. We should connect the microscope with a power source. If we use a mirror instead of an illuminator, this step can be skipped. Instead, you can find a place where the natural light is easily accessible. Step 2 is turn the roving nosepiece so that the lowest objective lens is in position. Once we hear the click sound of the lens, it means that the lens is in place and we should stop rotating. Step 3 is we should make sure the specimen is protected by a cover slip or a glass cover. Then mount the specimen onto the stage. Step 4. Use the stage clips to keep your slide in place. Make sure the slide is positioned centered under the objective lens. Step 5. Look into the eyepiece and slowly rotate the coarse adjustment knob to bring the specimen to focus. See to it that the slide does not touch the objective lens. Step 6. Adjust the condenser for the maximum amount of light. Since we are on the low power objective lens, we may have to decrease the illumination. Use the diaphragm under the stage to adjust. Step 7. Now slowly rotate the fine adjustment knob until you get a clearer image of the specimen. Then start to examine your specimen. Step 8. After finish the viewing with the lowest objective lens, switch to the medium objective and readjust the focus with the fine adjustment node. The last step of using a microscope is, after we have it focused, proceed to the high power. That's all from me. Thank you.